Okay, folks, while we kind of wait for the tech component to get sorted out here, thanks so much for tuning in. Oh, don't want to hear that. Okay, we're in business. Welcome, everybody. Thanks for tuning in this evening. My name is Ariel Firebaugh. I'm the Director of Scientific Engagement here at Blandy, and I'll be taking care of some of the behind-the-scenes work on Zoom today. A few quick housekeeping notes. So we've set up tonight's program as a Zoom webinar, which means that while our three speakers are presenting, you won't be able to turn on your camera or your microphone. So if you're eating dinner or talking to your dog or making funny faces, don't worry. We can't see you. You're not going to become Zoom famous. So no worries there. But you are welcome to put questions in the chat at any time. I'll be keeping an eye on that and bringing your comments and questions to the attention of our speakers this evening. And then at the very end of the program, once all three of our speakers have had a chance to present, I am going to have Zoom give you the power to turn on your camera and your microphone if you'd like to do that. Um, so that way, if you would like to reveal yourself and ask a question with your human face and human voice, you will have an opportunity to do that. But happy to take questions, comments, um, emojis in the chat at any time. I'll be keeping an eye on that. So as Zoom may or may not have told you when you joined the meeting, this program is going to be recorded, and it is also being live streamed to YouTube. So if something funny happens with your Zoom, or you hear from a friend who wanted to tune in, but their Zoom wasn't working, don't worry, you can catch it on Blandy's YouTube channel. So that will be available to you. Um, So, if you haven't heard, Blandy is not just a great place to come and walk your dog and enjoy the ginkgos. Blandy is the University of Virginia Field Research Station. And each year, we're host to a community of 20 to 30 students and scientists who all live and do research here, usually related to ecology or environmental science. And it's quite possible that if you are a Blandy regular, you've seen some of these Blandy scientists out in the field catching insects or measuring milkweed uh, out and about. Um, and one thing that I'm really excited about is Blandy has a big milestone that is coming up. It's our centennial. So the first Blandy students arrived here at 1928. And over the years, Blandy has been home to just generations of young scientists studying the natural world. So we're really excited to continue this legacy of being a place where students learn about the natural world and learn about uh, sharing their research with the public. With our programs tonight, featuring three amazing Blandy farmers. So we've got Willow Lovecky. She studies bees. So we're really working at that organismal end. We've got Stephanie Petrovick studying grassland communities. And we've got Kim Union, who studies different ways that ecologists can monitor vegetation across large scales. So we've really got a smorgasbord of ecological scales going on with our presenters tonight, the organismal, the community, and then the ecosystem landscape scales. So super excited to learn from our speakers this evening. I'm going to go ahead and pass the baton over to our first speaker, Willow. So Willow, the floor is yours. And folks who are tuning in, please feel free to drop questions, comments in the chat. I will be keeping an eye on that, and we'll save some time after Willow's talk to address some of your questions before we move on to Stephanie. So Willow, the floor is yours. Take it away.
All right. Thanks, Ariel. Hi, everyone. I am so excited to be here this evening and talk to you a little bit about my research. I'm going to be talking to you about bee and wasp nesting habits, talk to you a little bit about the different materials that they use, and so on. So first, I just want to start with an introduction. I'm someone who has been at Blandy for a pretty long time. I started here actually in high school, and I started volunteering at Blandy in high school. I grew up in Winchester, so Blandy's a really special place to me. After high school and volunteering at Blandy, I went to the University of Wisconsin-Madison, where I studied entomology and American Indian Studies. After I graduated, I did some work as a lab technician, an outdoor educator, and also an artist. And after that, I came back here to Blandy, and I am now a PhD candidate at UVA, and I work with Ty Ralston. So that's just a little bit about me. I'm really excited to be here tonight and tell you about the work that I'm doing. So starting off with some background, I first want to talk to you all about parental care. It's something that people probably know about too, and also a lot of different organisms spend a lot of time and energy caring for their young to play a role in their offspring success. And so parents dedicate time and energy to ensure that their young make it to adulthood. And generally, parental care is measured through behaviors like provisioning, so feeding offspring, and also incubating. So a lot of this work is we can think about birds. But one behavior that's really interesting to me is nest building. And so this is another parental behavior that is very costly to and in time and energy to the parent. And so I'm interested in thinking about this behavior. Who are building these nests and what are they building nests out of? And so first, I just want to talk about like why do organisms build nests? And so generally they're to provide shelter for their developing offspring. And this can be both abiotic things, so that can be, to regulate the temperature of their offspring or protect them from precipitation like rain or snow or different things like that. They can also be protection from biotic threats, and that's something that I'm really interested in. So protecting from predators and parasites. So that's a little bit about why organisms might build nests. But another thing I'm really, really interested in is materials that organisms use. And so to build homes and to build these structures that provide protection for offspring, organisms use all different types of materials to build these homes. And I like to draw attention to how people where this is something that isn't much different for us as humans. We also build our homes and houses, which are basically these places of shelter out of material from the natural landscape. And while when we look around at our home, the things that build the structure are a little bit more derived. They are the basic same like fundamental materials that other organisms use. And so some examples here, like a lot of different birds' nests, so things like wood and soil and also grasses, a lot of different muds. So like these nests down here in the corner, these are some wasps, which that soil is not very different than cob or adobe that's used in the Southwest. And also there's some really cool like leaf structures. These are built by ants mixing leaves and silk. And so just a pretty interesting part of nesting that I'm really interested in. And so I want to introduce you to some of the research questions that I'm interested in and some of the things that we'll talk about tonight. And I'm particularly interested in sort of why organisms build nests and also what materials that they use. And so particularly I'm interested in looking at this in bees and wasps. So first, I'm interested in what materials are used by bees and wasps, and we're going to talk about this quite a bit this evening. I'm really excited to show you all like the really weird and cool things that bees and wasps use to build their nests. Also how those materials that are different or similar to each other is something I'm really interested in. And also how different materials might affect parasitism. And we'll get into that a little bit at the end of my talk. But it's that kind of reason for why nesting provides shelter and protection from those biotic threats, so like predators and parasites. So I'm interested in doing this research in bees and wasps. And so to start off this introduction into the wonderful world of bees and wasps, I wanted to go through bee sociality, bee and wasp sociality, because oftentimes when we think about bees and wasps, we think about these very social organisms like bumblebees or like paper wasps. And so social species are individuals that have a queen. They have a queen and also workers and male. So they have this sort of caste system where there's a division of labor, and labor is divided among these castes to rear offspring. Bees and wasps also can be isolated. So they are individual mothers who do all of those sort of tasks on their own. They find nests, they bring in nesting material, they lay the eggs, and they provision bringing in food for the offspring. So they do that all by themselves without the help of workers. In the middle, there's these aggregating species, which are very similar to isolated. So they have individual mothers, but they nest in close proximity to each other. So that's something that's kind of interesting, is that even though they are individuals and they're still a lot next to each other, they don't have the queens or workers and those sort of things. And so you might have seen this example if you've been to the bee wall at Blandy. And so these are chimney bees. They're an organism that I'm really interested in. I'm not going to get into them too much this evening, unfortunately, but they are a very, very interesting organism that aggregates. And if you come in the early spring, like in May, you'll probably see the bee wall that's covered with all different types, not all different types, but a bunch of different individuals of this one species of bee that all nest close to each other. Before moving on, I just wanted to say I kind of presented these three different boxes that bees can be in, but it's actually like very much a spectrum, and you can have some species that are aggregating, then some years are social and some years aren't, and then also different species that sometimes in high elevations they're social, but in low elevations they're not. So these are bins that are really helpful to help us think about bee sociality and help us understand bee and wasp natural history, but there's also a lot of individuals who kind of can fluctuate between these three. And so my research is primarily interested on aggregating and isolated species. So that's what we call solitary bees and wasps. So the social species that you think of with bumblebees and paper wasps, they aren't really going to be who we're talking about this evening. We're going to switch focus to solitary, so these aggregating and isolated species. And they're really interesting. They actually make up almost 90% of bee and wasp species are solitary. So that's something they make up most of the bees and wasps that we see. And so they're really interesting to study. As far as getting into some of their nesting behavior, they generally have two different main modes of nesting. And so we can have ground nesters, which make up about 70% of species, where they are literally nesting in the soil. And then we also have cavity nesters, who make up around 30% of species. And so they nest in cavities that have already been established, and that could be by other organisms, or they can also be like by humans. So maybe you were building a deck or using wood, and you drilled a hole into that piece of wood, and you decided not to put anything in there. A bee might come or a wasp might come and find that cavity and decide it's a great spot to nest in. So they love those already kind of pre-established places. So now that we kind of know where bees and wasps might nest, I want to kind of get into the weeds of the structure of like what is actually going on inside of these nests, and what is some of the terminology that we use and some of the different structures that we can help think about the materials that are used. So this is an illustrated version of these two different kind of nesting types. So on the top we have cavity nesters, and again, this is an illustrated rendition, so there can be a lot of different variation in here, but this is just to help us get an idea of what's actually going on inside of these nests. So with the cavities, you have these sort of, long cavities, and then inside a bunch of different cells that the mother has created. And then in the ground, you have a cavity that's created by the mother. So she'll go in and dig into the ground and then create these series of brood cells inside of there for her offspring to nest in. And so that's there's a little bit of a structural difference and also a substrate difference. But now I just want to talk about some of the structural parts that are similar in these different nests. So first, as I kind of talked about, we have nesting substrate. And so nesting substrate is the area that's around the nest that's already pretty much established when the nest is started. And so this can include sort of those things that I talked about with cavity nesters. So that's like the hollowed out plant stems, dead wood, and also it can even be like snail shells. So there's lots of different things the nesting substrate can be for a cavity nester. For a ground nester, it's pretty straightforward. Soil is going to be that substrate. So she's going into the soil, and that's going to be the primary thing that's around her nest. We also have provisions. And so I mentioned this word earlier. Provisions are what's being put into the nest. And so with bees, that's going to be pollen. So that's all that yellow that's there. But for wasps, which we're also talking about this evening, they provision generally prey that they go out and collect. And that is very specific on the different types of species. And I'll talk about that a little bit later. But that can be everything from spiders, caterpillars, aphids, crickets, you name it. There's a wasp that's probably hunting that and bringing them into these nests that their offspring will feed on in there. Okay, next we have cell lining and cell walls. And so I put these two things into one slide because these two things are made by materials that the mother is going out and collecting and intentionally bringing into this space. And so that's something that I'm going to talk a little bit more detail about and something that I'm really personally interested in because I think it's super fascinating. So as far as cell linings, that's going to be the area that's directly exposed to the offspring. And usually there is well, I'll get into that a little bit later actually. So the cell lining is the area that's directly exposed to the offspring. And then the cell wall is going to be the area that divides those different brood cells. And that's pretty much always made by the mother. And so she'll bring in material and create that sort of wall that's in between each of these brood cells. So what types of materials do bees and wasps use? They use all kinds of things to create these linings and also these walls. And so there's a lot of different bees and wasps that use soils. That's a very common thing. That can be everything from rocks and pebbles to sands and clays and silt soils. And I'm going to show you a whole bunch of examples of these things. But there's also a ton of different plant materials that are used. And that's everything from plant resins, woody materials, plant wools, and also leaves. And then secretions are also really common, especially for that cell lining. So most of the time, especially bees that are nesting in the ground, the mothers create this lining that's on to help the humidity, control the humidity and moisture that's in these cells. So they bring in a lot of different things into these areas to ensure that their nests are secure and the offspring can successfully rear to adulthood. Okay, so now we're going to talk about some examples. We're going to actually look at some of the nests because they're really cool and really weird and super exciting to look at. So I'm just going to go through some examples that aren't necessarily in Virginia, and then we'll talk about some that are specifically at Blandy. But I think it would do you all a disservice to not talk about some of these weird nesting materials that are used. So first, got to talk about pebble bees. In the genus Dianthidium, there actually are some species that are here in Virginia, but they're really common in the Southwest. They use plant resin and pebbles. So kind of similarly to our brick houses, they have these pebbles, they bring them in and then they mortar them together using resin. So you can see all this gold that's in here is resin that she's gone out and collected. She creates this beautiful little brood cell. And then this hole that she's able to bring pollen in and other those different secretions of pollen, nectar, and then lay her little baby in there. She'll close off the top and then the baby bee will develop, eat all that pollen and make it to adulthood. So really cool, really weird, like it's super interesting that they use these materials. Next is another example of plant resins. So this is another genus of bee that's really closely related to Dianthidium. And they use plant resins and also plant hairs. So they create this sort of mixture of resins and hairs, mix them together into this sort of like really hard, sticky cell wall. And so they're actually not really a cavity nester I guess both of these aren't really cavity nesters or ground nesters. They kind of create this sort of in between. So that might have been a little bit of a confusing framework, but they create this wall out of this really crazy material right out in the open. So that's a really, really weird. So you can see like in here the different plant hairs and then the resins. And so she'll mix all those things together, create this really sturdy, really sticky, pretty safe nest for her offspring to develop in. More resins, because I think the resins are really interesting. We don't have a ton of them here, which is why I want to talk about some of like the weird combinations. But this is another genus of bee, Trachusa perdita, and they use plant resins and also leaf materials. So resins are really common in a lot of these species of solitary bee, and they're used in a variety of different ways. So again, this is kind of similar. So they are cavity nesters, and then inside of those cavities, they make these little brood cells, and the lining is out of this leaf material and resin kind of mixed together. And then inside of this cell would be where the pollen and the eggs are stored. My final example is a species of mason bee, Osmia ovaceta, who use flower petals. And it's really cute. And I feel like I couldn't talk about this without talking about them because they are very, very cute. They are actually from the Middle East. But we also have some species, actually, I think one species in the, I don't even know if it's in the Mid-Atlantic, but at least on the East Coast that also uses flower petals. But this is a very famous bee for the beautiful little brood cells that she makes out of these flower petals. So very, very pretty. Okay, so now we're going to talk about the bees and wasps that are at Blandy. So now that we know a little bit about nesting material, the different materials that are used, we're going to talk about the individual species and groups that I've found at Blandy and that I'm interested in studying. So going to kind of go back through these research questions to frame the rest of the talk that we have here in the next few minutes. I'm going to kind of run through here. This 15 minutes went by really fast. Okay, so first we kind of establish what materials, or we're going to talk about what materials are actually used by the bees and wasps that are at Blandy. And so to do this, I created these trap nests, put them out into the field to kind of get an inside of those trap nests. I can look into the nest, and I'll have some pictures up here soon to look at who's nesting in there and what materials they're using. So first up, we had quite a few mason bees at Blandy. You may see them, they come out in the very early spring, and they use soil and also masticated leaf material. So you can see up here all of these brood cells in this cavity, each of these individual little grubs, and then the mud that's used. And this bottom example, this is masticated leaves. So they actually go out, collect the leaves, they mash them up in their mouth, and then create those cell walls between each of these brood cells. Next up, we have leaf-cutting bees, and as their name suggests, they use leaves. So they have these really giant mandibles. They go out to different leaves and they cut sort of these circles and then very carefully lay them one another to create the lining and also cell walls inside of there. We do have some resin bees at Blandy. The one that I primarily saw is the giant resin bee, an invasive species. You might see her out and about in the sort of mid to late summer, very big bee. And they use plant resins, and it's a very sticky, very hard material. Pretty much as soon as they've nested in one of these nests, you can't get the material out. It's very, very hard, very protective, and it's one material I'm particularly interested in. So that's you can kind of see the sticky and then all within that nest, and then you can see the yellow, each of those brood cells. So you have like one here in the back, four on this bottom one. You can see the yellow pollen that she's put in there for her offspring. Next, we have the wool carder bees. These guys are also an invasive species that's at Blandy, but they're really interesting. When I first opened up this nest and they were in there, I had no idea who was nesting in there. I was like, what is this weird sort of like spidery material? But no, it's actually like trichomes, so the little plant hairs. And these bottom pictures I put in here about how they kind of showing how they collect it. So they cut off all those trichomes and roll them up, and then they use that as their nesting material. So this very sort of fuzzy, nesting material that's all around them. And you can kind of see the rounded shapes of each of those brood cells in there. Moving into some of the wasps, we have the grass-carrying wasp, the most common species that I saw in my trap nest at Blandy. They hunt katydids and tree crickets, so you can see those guys stuffed in there. Those are all of the prey that she brought in there for her offspring to feed on. So you can see the little baby wasp that's in here. And then the material that she uses is actually grass. So she cuts up all these little pieces of dried grass and very carefully places them to create these little cell walls. And you can actually see in this bottom picture one of the mamas who's in there, and she's nesting right as I took that picture. We also have mason wasps. So similarly to the mason bees, they use soils and muds to create their cell walls and cell linings. And so they actually hunt caterpillars. You can see each of these cells packed to the brim with these green caterpillars. So many of them. Very, very cool. And so from this, we can see all these different types of materials. So lots of different plant materials here at Blandy. We have the grasses, the masticated leaves, the big full leaf particles, the plant resins and plant hairs, and then all these different types of soils. I didn't actually get a chance to show all of them, but lots of different soils that are here. And so that kind of establishes my first research question of what materials are used by bees and wasps. So we see all those different types. And so that leads me to the second question, which is about how the materials are different or similar to each other. And this is something that I'm currently working on. And so to do this, I'm collecting nesting material again this summer. I'm putting more trap nests out at Blandy, and then I'm going to collect those. And what I'm going to do is actually measure thickness of all of these cell walls. And then I've been working with people down here in Charlottesville at UVA in the structural engineering department and material sciences. And we actually figured out a way that I can measure compaction. So it's using this hand crank machine. And what I'll do is take each of these cell walls and put it in there and then actually be able to measure the amount of force that it takes to break through each of those nests. So I'll have a way of quantifying how strong they are and comparing them to each other, which is really exciting work. And stay tuned because I'll be very excited to tell you all about it. Okay, so we got question one, question two, and then finally, my third question is just about parasitism. And I'm not really going to get too into this, but I think it kind of will be able to shed a little bit of light on why these questions are interesting. Why am I interested in comparing the different materials? And so parasitism is actually something that we see in all these different species of bees and wasps. And so what we have is a host species and then a parasite. And so parasites are pretty much dependent on the hosts for their success. So they usually, and there's all different types of natural histories, and I could spend another 20 minutes just talking on that, probably even longer. So definitely ask questions about parasites if you have any. And so their success as parasites is also, I'm interested in seeing how that's dependent on the different materials. And so I'm just kind of going to walk through a quick example to show why that might be. And so we have these different types of nests, right? And so one thing that I'm interested in thinking about is how the structure and material might influence parasite success. And so that's going to depend on this parasite life history. And so I'm just going to kind of walk through this. So you might have a parasite who they actually lay their egg while the nest is open. So while that individual mama bee is out collecting pollen, collecting all these nesting materials, she's leaving her nest wide open. And so these flies will come in and actually lay their eggs while she's gone and then leave. And then maybe that mama bee or wasp doesn't realize that the egg is in there and she seals it up with that parasite egg in there. Another example is that the parasite might actually be able to lay the egg through the material. And so she'll lay that egg oftentimes using an ovipositor that has sort of sharp serrades on it, and then go through the material, lay the egg, and then that egg is in there to develop. And so with the first example, that mama parasite did not have to deal with any nesting material or nesting structure, but the second one did. And regardless of how the eggs actually get in there, both of those are going to become adults and have to get out somehow. And so that's one of the things I'm really interested in understanding, and one of the reasons why it's super interesting to think about why these different materials are used. So with that, I think that will conclude my talk, and I think we'll do questions at the end. So I can pass it over to Ariel to pass it over to Stephanie. So thank you
Okay.
guys.
Thanks so much, Willow. Great talk. Hey, folks, you're welcome to put questions in the chat at any time. Um, oh, Sean's got one already. Is there any collaborative work with material scientists? Seems like material scientists could learn from how bees are using all of these diverse materials. Historically, the honeycomb structure has had obvious benefits to construction materials, as well as applications where strong but lightweight is important, like airplanes, boats, etc. So can you speak to any of that, Willow?
Yeah, I think it's been really fun collaborating with people in material sciences. I've learned a lot because I was kind of like, okay, I have all these materials. I'm really interested in comparing them, but I have no idea how to. And so then I went to all these material scientists and I was like, how do you guys compare materials? And so they had a lot of insight. And I think they're all kind of interested in seeing like especially the resins. That seems to be one thing that everyone's interested in is like how different are these resins because they're so strong and anytime we get them in nests, like it's impossible to get them out. So I have heard about like some of the structure stuff like with honeycomb and that design, but I'm not as sure about like these materials that I talked about today, besides the fact that I think humans have also kind of established and found materials that bees also use and we use in our homes too. But yeah, it'd be cool to see how we might learn from them moving into the future.
I can speak to the strength of that resin material. When we have
It's
nests
crazy.
with those bees, you can't hardly pry the little blocks apart. They're just stuck on there so good.
Very strong.
Um, okay, last chance for questions for Willow for right now. We'll save more time at the end. But I've got a question for you, Willow. Favorite bee, least favorite bee.
Oh man. I don't know if I have a least favorite. Let me think. Okay, favorite, I actually didn't talk about in my talk, but my entire dissertation is about the chimney bee, which is the one of the bee walls. So if you want to learn more about my favorite bee, you should come out to the bee wall in May because I will be there the entirety of May and I can tell you all about chimney bee. Least favorite bee, I mean, it's hard because I mean, you might say the giant resin bee because the giant resin bee is crazy. They, one, bring all that resin into the trap nest, which is like really hard to clean out, and they're also invasive. But they also use the resin as a defense mechanism, and they will spit it onto carpenter bees and prevent them from flying, which is kind of evil and maybe a little mean. So maybe, I mean, I still think they're really cool, but maybe more on the least favorite side of the bees here.
Fair enough. You heard it here first. Well, thanks, Willow. Folks, if you think of a question for Willow, feel free to type it into the chat. I'll be keeping an eye on that. Willow can keep an eye on that too. But I think we are going to go ahead and pass the baton over to our community ecologist, Stephanie, who will
Hello.
be sharing all about grassland communities and her work at Blandy. Stephanie, the floor is yours.
Thank you, Ariel. Let me get this shared and set up with everybody so we can get started. Here we go. Okay. So, hi, I'm Stephanie. So I'm going to talk a bit today about how nutrients and disturbance shape grasslands with a specific focus on humans and their activity as well as invasive plant species. And so, a bit about me. I grew up in Massachusetts, and then I went to the University of New Hampshire and did my bachelor in environmental science. And there I focused on forest-related research. And then I got a really great opportunity to kind of come to the University of Virginia and do my PhD here in environmental science. And I did a little bit of a shift in my focus, and so now I focus on grasslands rather than forests. And though I'm always happy to talk about both. And that is how I ended up at Blandy doing research in the successional grasslands there. And so today I'm going to sort of go through some topics here. So I'm going to start with grassland dynamics. So how do these plant species interact with each other? How do we get the plant community that you see when you are out walking around at Blandy? And I'm also going to talk about the impact of human activity, as well as how invasive and non-native plant species play a role. And then I'm going to talk about what it all specifically looks like at Blandy. So what do I do at Blandy um, in regards to these topics? And so I'm going to start off with, well, why is this important? Why do we even care about these grasslands beyond how absolutely pretty they can look? Well, they're actually pretty useful to humans. We use them a lot for grazing, greenhouse gas regulation, they're very helpful for soil formation. There's also a lot of use for ecotourism and public use because, as we can see, they can often be very pretty to look at. And they're also very key sources of biodiversity since they serve as the habitat for various species. And unfortunately, these grasslands are often at high risk from humans because, well, humans often take grasslands and convert them to different uses. For example, it's a lot easier to build somewhere if you don't have to chop down a whole bunch of trees, right? And so, while many grasslands are lost to these sorts of human activities, there's a lot that also get regained. For example, many abandoned agricultural fields end up returning to a more natural grassland state over time. And so this has led to a lot of questions about how are these grasslands going to now develop? Are they going to differ from what was there before humans interfered? And how can we best help these grasslands sort of return to their former glory? And so this is the grassland I work in at Blandy. And within this grassland, I study plant communities as well as competition between these plant species and the impact of nutrient addition and disturbance on these plant communities. And so it's important to understand this research by starting with how plant communities will take shape. And so to start, after species are introduced to an area, their success are going to depend on a few different factors. So first, we have habitat size and suitability, the number of individuals of that species that are present, as well as how well these individuals established themselves and who among these species even arrived first, because arriving first gives you a big advantage. We also have resource supply as a factor, as well as interactions with other species, such as predation/herbivory and competition with other plants. And of course, there is a healthy dose of luck involved. And so what I like to focus on are the resource supply and interactions with other species. And so that's what we're going to talk about today. So these plant species are going to be limited by one or more of the resources in shortest supply. This can include light, carbon dioxide, water, nutrients, any of these glorious things that help plants grow, right? And if multiple plant species are competing with each other, the plant species that can survive at the lowest resource level for whatever resource is limiting in the system is then going to become the most common dominant species within the system, right? And so that's how you get, a grassland where you're looking and it's like, oh, it's all the same species of grass, right? And so to sort of deal with this competition, all of these plants that want the same resources end up adapting different mechanisms to take them in, and that is how we get sort of these balanced communities that we can also see. And it's why you don't just walk somewhere in nature and only see one species. There's usually a lot of different species present, even if the amount of them varies, right? However, humans alter these dynamics. We add nutrients to systems. We see nutrients, for example, run off from agricultural fields into the surrounding areas. We see disturbance from mowing and tilling and all these activities, and these have an impact on these plants. And nutrient addition specifically is going to remove limitation from the resource supply, right? So if it's a system where it was previously limited by the amount of nitrogen available, well, now it's got all this extra nitrogen from human fertilizers, right? And so that's going to change things. And in grasslands specifically, we see increases in plant productivity and growth. We see reductions in plant species diversity, as well as an increased shift to a new species composition. So species that were present before may now be replaced by other species, right? And we can also see a reduction in the number of factors determining where the different species can live, which is what we refer to as the niche. And this becomes even more complicated when we take into account invasive and non-native species. And so communities that in nature have a higher native diversity have less invasive plant species present. There's just no room for them, right? The plants already present have already taken up all the resource supply, and there's nowhere these species can interject themselves. However, when we add nutrients, this balance changes. We see decreased diversity of native species under nutrient addition, and this allows for an opportunity for these invasive and non-native species to really enter the system. And we also see a change in conditions where we see increased nutrient availability, right? And this can be more favorable for non-natives as well and invasives if they can survive at lower levels of these nutrients. And so we see that there is an increase in non-native and invasive species and a decrease in native species in response to nutrient addition. And then the question is, well, what about disturbance, right? So disturbance often takes the form of land use change. Most commonly, we see mowing and tilling, such as for agriculture, but we also see land use by humans in the forms of grazing and infrastructure, such as roads or buildings. And so disturbances, such as mowing and tilling in particular, have been shown to increase species diversity. And well, why does that happen? Well, because there's less competition. You've kind of removed everything that was previously there, and so now it's a level playing field, right? And this, again, has implications for invasive species. Disturbances change the conditions that are present. We've reduced things in the example of mowing and tilling to basically bare soil. This means that there's more light available and there is less competition from especially native species. And these invasives can take advantage of this with various adaptations that they have that sort of benefit them within entering these systems. For example, they can have more efficient seed dispersal than native species, maybe a quicker growth rate. Maybe they even have a larger range of optimal conditions, right? And there's many more adaptations that they have, but those are some big ones. And so what we end up seeing is a higher diversity and abundance of these non-native and invasive species than any native plant species after these disturbances. And combined with nutrient addition, that can make it even better for invasive species. They love that, right? And so it's a bit complicated when we're looking at these grasslands that are recovering from these disturbance and nutrient addition activities. And so I am sort of investigating this at Blandy. Like, if we sort of look at these disturbance and nutrient addition treatments, do we see these dynamics, right? Is the plant community changing in response to nutrient addition? Are species becoming more or less dominant than other species? And does this relate to their status as non-native versus native, or maybe are they invasive? And how does the plant community respond to disturbance? Do some species succeed better in an area that was disturbed than not? And is that influenced by their status as native or non-native? And so at Blandy, I have 25 plots that I've set up. And across these plots, I have 4 treatments that I want to look at. So I have nutrient addition, where I have added nitrogen, potassium, and phosphorus fertilizers. And then I have disturbance treatments, where I've gone through and mowed and tilled these sites. I also have a combined nutrient addition and disturbance treatment. And then, of course, to compare to a baseline natural state, I have what we call the control treatment, where nothing has been changed. And so, in order to sort of answer this question of native versus non-native, I take measurements of the plant community. I take a whole ton of other measurements in this system as well, but for these specific questions, I focus on my data that relates to plant species presence and abundance. And so that takes the form of percent cover measurements. And that basically means how much of a measurement area contains a specific plant species. And this is sort of a visual estimate that we do in like a huddled group, like in the picture on the slide. So from all of this data, I have found at least some patterns. Not all species show relationships, but some of them do. And so the non-native plants, spiny plumeless thistle and great mullein, which we can see in the two tall pictures on the left bottom of the screen, prefer any plots where disturbance occurred. This can be disturbance with or without nutrient addition. They just love disturbed plots. And meanwhile, the native ground cherry that we deal with, which we could see in a picture on the bottom right, will prefer any undisturbed plots. This includes control plots as well as the nutrient addition plots, where disturbance did not occur. However, this changes a little after 3 years of these disturbance and nutrient addition treatments being applied. We see that the native species, common milkweed, which we can see in the plant picture on the left there, will prefer any plots where there is nutrient addition to any plots where disturbance occurred. This kind of makes sense. They can take a little longer to grow and all these things. And the non-native species, yellow toadflax, is a little bit of a curveball because it shows more preference for control treatments than any disturbance treatments. And so for whatever reason, it doesn't really like these disturbed plots, which may have to do with its growth period and patterns there. And finally, we see that, as in year 1, common mullein still prefers disturbed plots, and spiny plumeless thistle, while it does like disturbed plots, it prefers plots where there is both nutrient addition and disturbance. So these nutrients are kind of coming into play now. And again, both those species are non-native, and so we see this change in dynamics. And we can actually see this on the ground when we're just walking around. And so on the left here, I have a picture of a disturbed plot. And so you can see it's all these very like leafy looking plants. So that is the common mullein in a sort of young stage of its growth. And if you see anything kind of spiny, that's going to be the spiny plumeless thistle, again, in a young stage of growth. And meanwhile, on the right side of the screen, we see just the base sort of control plot, where, you know, there's just all these grass species and there's some other species that would be present as well, but what we can predominantly see visually is the grasses. So very different looking. And so in conclusion, we can see, okay, nutrient addition and disturbance may be decreasing the diversity of native plant species, which, you know, we saw supported in previous research. And the disturbance seems to be increasing the presence of many non-native species. And combining with nutrients may also do this, or in fact, create even more incentive for some of these non-native species. And we also see that time and repetition of these treatments are factors. We saw a shift in what species were responding to the treatments after multiple years of the treatments being applied. And overall, over time, this is all going to probably result in changes in the plant community composition within the grassland, particularly where these treatments are applied. And so that's it. Do we have any big questions? And I'm happy to hand things back to Ariel now.
Oh, thanks so much, Stephanie. Sean coming in again with a question right away. Is it realistic to attempt to sort of re-baseline the soil, maybe even just on a smaller scale, like a large conservation area, so that the soil will have compositions like they may have had before human intervention changed it significantly? Do we even have data to do that rebaseline? So maybe thinking about, what did the soil look like before humans came onto the scene? How is there a way that we could figure that out? And thoughts about restoring the soil to that point.
Yeah, so I think that would depend just system to system, sort of area to area. For example, if you have an area of grassland where only part of it was affected by human activity, whereas the rest remained a natural system, you can probably compare the soil within these two areas and get a pretty good picture of what it would have looked like before humans sort of came into the system and changed things. But in a lot of areas, it's really, really hard to even get that sort of picture. Because no one was taking soil measurements before humans came in and hung out and did whatever they wanted with what was going on. And that's actually a problem in a lot of areas where it's like, well, even if it looks like a very natural system now, you can look at land use records and it's like, oh, 50 years ago, that was an agricultural field. And so even if you think it's going to be an untouched soil, sometimes it turns out it isn't. And so in my system, there's been the last time there was any sort of disturbance or activity was in, I believe, 2002. And so I kind of just have to go with, okay, it's been like about 20 years, 18 years before any sort of further activity occurred. And so I just have to treat that as some sort of baseline, unfortunately. But if I can ever find a very similar system nearby that didn't get disturbed, that would be great. I would love that for my research.
That's
Um,
great, Stephanie.
yeah.
Have you considered buying a TARDIS with your next grant?
Oh gosh, if only, if only the having a time machine go back like the TARDIS. Oh, I could take so many fun samples. I would love that. That would be fantastic.
One more question,
Mhm.
and folks, feel free to put questions in the chat at any time. We can get those addressed. Um, but your favorite plant in your plots at Blandy
Mhm.
and your least favorite plant.
Oh boy. Okay. I think my favorite one is probably that's so much harder. I really like the common mullein because it's really fun. Because it's like you might remember from the picture, it ends up doing this stalk and it has these really pretty little yellow flowers at the top and then the leaves at the base and everything. It's just very fuzzy to the touch, and that's just so fun to interact with. Um, and then we also have catnip out there, which so fun. Absolutely throws everyone for a loop whenever you start smelling mint when you're out there. But it's just the catnip, right? So that's also very fun, especially because it's structurally very different looking from a lot of other plants. Even though it doesn't seem like that if you just glance at it. So very, very fun plants there. My least favorite is definitely the thistle. I hate it with a burning passion because it has a tendency to take over plots and it hurts so bad to work with it because it has all these little spines on it, which is where spiny comes from in the name. And so it's just a pain. Hate it. Could do without.
I thought you might say that.
Yes.
So, folks on the Zoom, next time you come to Blandy, go to the succession fields, look for mullein and thistle, and think of Stephanie. And maybe look for Stephanie too if you're coming during the summer. Okay, thanks so much, Stephanie. Um, you can help me keep an eye on the chat if people think of questions for you. And last but not least, we've got our landscape level ecologist, Kim. Kim, the floor is yours. Science us.
Hi, is that full screen or no? It doesn't seem like it is on my screen.
It is almost full screen. The slides look pretty big and we can just see the shell around it a little
All
bit.
right, that's good enough for me. Okay, so yeah, my name is Kim Union. I'm a PhD student here at UVA Blandy as well. I don't have an intro slide, but I was at NYU in New York before this, and I also grew up in Massachusetts like Stephanie, and I think being in the city, I was studying environmental studies, and I realized that I just wanted to be spending more time outside and that I really missed being in nature and, yeah, working outside, being in the sun, and all of that. So, coming to UVA and having a field component to my research, especially at a place like Blandy, where we're so free to just be in the environment and figure out what we want to do with our like time while we're here and doing field work is so feels so great and like so just like a great way to like we're getting our degrees and we're getting paid to play outside all summer. So it's a really great time here. Um and yeah, so my presentation is planes, drones, and automobiles. It's remote sensing and vegetation at Blandy. And I'll get into what remote sensing means and what like types of vegetation I'm talking about. Um, oh, are the slides going for you guys? They're not.
I'm still looking at your first
Oh,
slide,
there we go.
Kim. Now
Okay,
there's your second one.
sorry. Sorry about that. So just for some background, about like the big global and landscape level questions that I'm interested in, as opposed to like the organismal life history, community ecology. I'm interested in more about like global carbon cycling and how um, our total estimates of what carbon there is on the ground are super uncertain, and we have different estimates coming from different methods. So when you're looking at remote sensing data, so data that isn't directly collected in the field, um, you get really different results as opposed to when you're looking at like field and then scaling up. And you can see like the land CO2 sink here. We have a bunch of different lines, and those are when we're looking at models or we're looking at like satellite estimates. Like you get predictions and estimates that look super different based on how you're calculating the amount of carbon in a system, and therefore the amount of carbon sequestration that's happening when vegetation is growing more in warmer temperatures and other impacts of climate change. So understanding how vegetation is growing and changing is really important. And currently, our estimates are a little unclear and they don't always line up. So Stephanie already talked kind of about why grasslands are important and why these kinds of systems are important. So I'll skip over this one, but I'll get to remote sensing. So I've said it a couple of times, and just if you're not familiar with that as like a term, it just mostly means any time where you're measuring without some sort of like invasive or like direct sampling. This can be like images or this could be like taking a picture of something with a camera or a picture from a satellite or some other kind of imagery from a satellite. It can also be something like an aerial, like a plane and flying over and collecting data, or a drone collecting data, and it can be like on the ground collecting data where you're not physically sampling, which I'm still taking physical samples, but then I'm comparing it to these remote sensing data to see how well the remote sensing data is doing and if it's actually accurate compared to the physical samples. So that in the future, we might not need to take physical samples at every location, and we can just rely on these remote sensing products. So when I'm talking about vegetation, for me, what that means, one big part of my research at least, is non-photosynthetic vegetation. So I just want to define that now. I'm going to call it NPV a good amount, but that just means anything that is not photosynthesizing, like just what it sounds like. So things that are standing dead, so they're not alive anymore, but they once were, or they're litter, and so they've like fallen down and they're horizontal. So just stuff that either was initially structural, so it was never meant to photosynthesize, or stuff that has died and therefore is no longer performing photosynthesis. So for me, when I'm looking at vegetation, I'm mostly looking at, yeah, large amounts of vegetation as my big question. So I'm just breaking it into photosynthetic vegetation, anything green and photosynthesizing, wood, litter, which for me means anything dead and on the ground and like horizontal, and standing dead, which means some stuff that is upright, things like stems and leaves that are still attached, but they have died or senesced. And the reason that I'm interested in NPV specifically is because it doesn't show up the same way that green vegetation does in remote sensing data. So a lot of remote sensing measures greenness. So when you're comparing a green leafy plant to wood, it's not going to look the same because you're looking at greenness. Or if you're looking at trying to look at total like mass or like amount of vegetation and they're looking at height, you're still not really getting the same data from a tall alive tree versus like a fallen log. So you're missing a lot of the important data when you're only considering one variable to measure vegetation. So if you're only considering like height or canopy height, which is a really common forest metric, or you're looking at greenness, you're still missing a lot of the vegetation that's there, if that's what you're using to calculate how much is on the ground. So just to like further on how much you might be missing when you're just calculating, looking at green systems, a lot of systems can contain more or as much dead vegetation as live. And we don't really have good estimates of NPV across heterogeneous systems. So there is a lot of work on crop fields looking at non-photosynthetic vegetation, looking at crop residues like after harvest or after clearing. But in systems where you have this like mix between green and non-green vegetation or in between seasons or where there's multiple types of vegetation, there isn't a lot of good work currently. There's a lot of like emerging work, but there's not a lot of established work kind of able to parse that difference between green and non-green or photosynthetic and non-photosynthetic. And so one thing we need when we're looking at these like larger scales is improvement across and between scales from the field level to this all the way to the satellite level. And we need specificity within NPV. So not just saying like, okay, here's everything photosynthetic, here's everything non, but like once you get to non-photosynthetic, what is the difference between that wood, that standing dead, and that litter? Because we're not distinguishing those when we're trying to calculate our total carbon on the ground. And this is important because it can help us track crop residues, like I said, but also if pastures are being degraded, if there's a change in wildlife grazing, and something important like fire risk or insect outbreaks and diseases. If we're constantly monitoring a system and we're seeing differences in the amounts and the types of non-photosynthetic vegetation, that tells us that something like a higher fire risk might be occurring, or there might have been an insect outbreak, or any of these other environmental shifts that we're not getting if the total amount of green vegetation is staying the same, or if we're just seeing, okay, green vegetation is decreasing, but we don't know why, and something like, is all vegetation decreasing, or is it becoming more non-photosynthetic outside of when we normally expect? That can all kind of indicate to us, like, is it because of an insect outbreak? Is it because of a fire? Is it because of grazing or something? So one tool, So I've established we have these big questions. We need better NPV quantification. Remote sensing is a way you do that. And one type of data that remote sensing tools like field, plane, and satellites can all give us is hyperspectral data. So a regular camera will have 3 colors. A hyperspectral camera is going to measure 100 different types of light. So it'll have 100 types of blue, 100 types of green, 100 types of red, 100 types of near infrared in comparison to that typical camera that has red, green, and blue. And an example of this is like a multispectral comet existing satellite. So NASA's Landsat satellite isn't just a typical camera, but it also only measures about 6 types of light. So it measures a few values, and then a hyperspectral imager at the same plot will have, maybe 200 to 2,000 different wavelengths of light being measured. And so we get all of these bumps and different like spectral signatures. We get this spectral curve, which is what the plot on the right is called. And it gives us all this information about what is on the surface beyond just what a few different types of light can tell us, but how the surface interacts with every type of incoming light. So a typical day for me when I'm thinking about hyperspectral imagery is going to be going out into the field and doing field spectroscopy. So I have this little, the SBC, this is ours. I don't have a picture of me in it, but it comes in this little backpack carrying case you can put it in. And then there's a handheld attachment. It looks like a little Ghostbusters like backpack with the gun. And you point it at a surface that you know how it reflects light, so like a pure white surface, and then you point it at the target vegetation. And then because you've adjusted it to how much light is coming in, it can tell you how much light the surface is reflecting. And I'm driving around in the Gator and doing these scans all across all the different vegetation at Blandy. And what this looks like after I've collected the data and once I'm analyzing it is you can see that these are two curves of each vegetation type, and you can see that like some litter and non-photosynthetic vegetation looks one way and green vegetation looks a different way. So we can tell a difference between both non-photosynthetic vegetation and photosynthetic vegetation, and between different types of non-photosynthetic vegetation. And so there's upcoming satellites and an existing European Space Agency satellite, which is EMIT, and then NASA's upcoming satellite, SBG, which will have hyperspectral capabilities. So once we understand the relationships between vegetation and spectra in the field, because right now we have maybe an understanding of how it is in the lab, but not a great understanding of it in the field for non-photosynthetic vegetation. Once we can really establish those relationships between the spectral curves that we're getting from our scans and the amount of vegetation and different types of vegetation on the ground, we can apply this to these big 30-meter pixels of global coverage that we're going to get from satellite imagery. And then this is an image of a different type of data that I've been taking at Blandy. This is with help from Robin Kim in civil engineering at UVA, and he flew a LiDAR drone at Blandy this summer, and that is that drone flight. And the thing about LiDAR is that it sends a pulse out in the form of a laser, and it gets it back, and that allows it to get this 3D data. And so yeah, so we get this 3D data both from the drone level, but also from things like NEON, which is the National Ecological Observatory Network. And Blandy is one of the 80 NEON sites, which are all across the US and funded by the NSF. And they have these low-flying planes that collect both the hyperspectral data I was talking about and the 3D LiDAR data I was talking about. So we can see at about every meter, they have the 3D image of what's on the ground, and they have a hyperspectral signature. So we can get all of this different data at Blandy about both what is on the surface from the spectra and how much of it from the 3D LiDAR data, which is really cool. And then you don't have to totally look at this. It's just a kind of conceptual idea of what I'm going to be doing, but you don't have to hear about is I'm going to be looking at all these different data sources. So just the drone LiDAR with the field spectroscopy, the neon spectroscopy, and the neon spectroscopy and the LiDAR, and all these different combinations to try and determine what data inputs are going to provide the best estimates of vegetation and types of vegetation on the ground. And yeah, that's it for me. Thank you guys, and thanks to specifically my two field techs, Alana and Caroline, because they were really great in the last 2 summers. A few questions for them.
Thanks so much, Kim.
Yeah.
Okay, I've got one for you.
Mhm.
Favorite data set to work with? Least favorite data set to work with?
I will say my favorite data set is always going to be the data that I'm collecting. So my field spec data, I would say, is personally my favorite because I can see the spectral curve, and then I also remember what it was like to be at that exact plot, maybe for like an hour at least, because I was in there sorting through the data myself. So I feel like it's really easy for me to visualize what it's supposed to look like and like get a sense for like, does this look right? Is something off? Because I know what was on the ground and on the surface. And I'm a very like visual person, and I think with remote sensing data, it can be super abstract. So it's really helpful to have been there. And for that reason, I would say my least favorite data to work with, I don't know. I think it might be the satellite data just because it's not at such a small scale, and a lot of the satellites that I'm interested in just don't have as good temporal coverage, which is why like it's exciting that there's all these satellites coming up. But a lot of the existing data, there isn't good temporal coverage. So there's maybe like, by that I mean like there's maybe like 1 day of clean data with the type that I'm looking for in the spot that I'm looking for. And I can't really picture it because it's just so large scale, and I think it's harder for me to really visualize. But that is like the exciting thing about the future satellites coming out.
That's awesome. Thanks. Well, virtual audience, I'm going to go ahead. We've heard from our 3 speakers. Thank you so much. I'm going to go ahead and stop the live stream.